Welcome to the Explainer, everyone. Let's just jump right in, shall we? Today, we're unpacking the absolutely fascinating, millennium-spanning history of how humanity went from sheer guesswork to modern antimicrobial medicine. We're talking about a wild journey of exploration, treachery, science, and a few accidental miracles that completely changed the course of human history.
Have you ever stopped to think about how we fought off deadly infections before antibiotics even existed? It's a reality we rarely have to face today. But for thousands of years, fighting disease wasn't exactly a science. It was mostly a desperate mix of lucky accidents and some truly bizarre potions.
Section 1. The Ancient Shotgun Approach, throwing everything at the wall. Back in the day, humans essentially just threw everything at the wall, hoping something, anything, would stick. For example, the Egyptians used a poultice made of grease, honey, and bits of linen. And they were actually kinda onto something, because honey does have natural antimicrobial properties. Over in the Middle East, they used the sap of the balsam tree to try and prevent infections. But to really grasp the scale of this history, we have to zoom out. We're talking about a timeline stretching from 1200 BCE, where folks were just throwing these wild concoctions at their ailments, all the way to the 1500s and 1600s, before we finally stumbled upon our very first targeted cures. For centuries, our best defense was literally just a guessing game.
And that brings us to the ultimate guessing game, a potion called Theriac. Around 120 BCE, King Mithridates VI desperately wanted a universal antidote to poison. He tested on prisoners, mixing up to 100 different ingredients. We're talking toxic plants, opium, and even the flesh of venomous reptiles. The Greeks helped spread its reputation, and eventually, just mixing up a batch of this stuff became a massive community festival. For nearly 2,000 years, Theriac was the absolute go-to drug. But here's the kicker. Despite its incredible placebo-driven popularity, it completely failed to stop actual infectious diseases like smallpox, typhoid, or the bubonic plague.
Section 2. Quinine and the holy bark, the first real cure. So, moving past those ancient placebos, our story of a real, tangible cure actually starts in the Andes with the Quechua people. They lived closely with the land and developed an herbal remedy using the bark of a tree they called quina quina, or holy bark. Now, they used this bark non-specifically, kind of like how you or I might take an aspirin to treat the chills of a fever. But when the Spanish arrived in the 1530s and malaria began ravaging their population, the bark's true specific power was finally revealed. Legend actually has it that it famously cured a Spanish countess, Ana del Cinchon. In fact, the scientific name of the tree, cinchona, is named after her. But the drug had a pretty rocky start in Europe at first. It didn't dissolve well in water, and fraud was everywhere. It wasn't until the 1670s when a man named Robert Talbot came along with a secret recipe. He brilliantly mixed the bark with wine, using the alcohol to perfectly dissolve the active ingredient, and he used it to cure King Charles II of England of malaria. A few years later, he cured the son of King Louis XIV of France. With these high-profile royal success stories, cinchona bark became world-famous overnight. It was so crucial for keeping workers healthy in malaria-infested regions during the Industrial Revolution that this bark literally became more valuable than gold. Now here's why this is such a huge deal. It perfectly illustrates the leap from ancient guesswork to actual targeted medicine. We now know that the active alkaloid in the bark, quinine, works by interfering with the malarial parasite's ability to digest hemoglobin. So even though the parasite is sitting right there in the middle of a red blood cell, completely surrounded by hemoglobin, the drug effectively starves it to death. Finding this fortuitous mechanism was just a monumental leap for humanity, an oral medication that actually eradicated a specific disease.
Section 3. The magic of immune serum, borrowing defenses.
Let's fast forward a bit to the late 19th and early 20th century to see how this builds. Science is really advancing now, and researchers are starting to look at our own bodies for the answers. Enter the idea of passive immunity. This is where you take the serum, which is the watery fluid of the blood, from a patient who has already recovered from a disease, and you give it to someone who is currently acutely ill. In the 1890s, scientists achieved a dramatic breakthrough using this exact method to treat diphtheria. Diphtheria was a horrific disease. People called it the strangler because it formed this thick mass that blocked the airway, and it carried a mortality rate of up to 20%. Injecting these patients with immune serum saved countless lives and actually won Emil von Behring the very first Nobel Prize in medicine. But this breakthrough created a massive divide in the medical community. Researchers were basically split into two camps. On one side, you had folks focusing on chemical poisons, trying to kill the microbe directly. The huge problem there was that at the time, these chemicals almost always caused severe, sometimes fatal damage to the patient too. On the other side, you had natural immune defenses, like serum. Serum was much safer and highly specific to the disease. But it was a logistical nightmare. It had a terribly short shelf life, required recent blood donors, and was incredibly difficult to transport safely before modern refrigeration existed. Section 4. Dying to kill bacteria. The chemical Trojan horse. Because of all those logistical headaches with serum, a frustrating but necessary shift back toward chemicals had to happen. And surprisingly, the big breakthrough wouldn't come from traditional medicine at all, but from the industrial dye industry. In the 1880s, researchers noticed that aniline dyes could actually stain living bacterial tissue. This sparked a brilliant concept known as the Trojan horse theory. The idea was simple, but genius. If a dye can specifically target and enter a bacteria, what if we attached a poison to the dye molecule? The dye would carry the poison directly into the microbe, killing it, without harming the rest of the patient's body. In 1910, Paul Ehrlich successfully used this concept to create Selvarsen, an arsenic-laced drug that treated syphilis. But it was still highly toxic, incredibly volatile, and it only worked on that one specific organism. The limited success of Solvarsan kicked off an absolute agonizing grind of trial and error. The German chemical giant IG Farben decided to just throw their massive resources at the problem. They shifted from those toxic aniline dyes to less toxic azo dyes and began methodically attaching different chemical chains to them. It took 4 years of incredibly tedious, mind-numbing work. They produced and tested over 3,000 unique chemically modified compounds.
The testing itself fell to a physician-turned-researcher named Dr. Gerhard Domach. Picture this. He's injecting mice with a highly virulent, 100% lethal strain of Streptococcus, giving half of them a new dye compound and just waiting. For years, all he saw were cages of dead mice, until one extraordinary day. Domach walked into the lab to find one cage of dead mice, just as expected, but right next to it, a cage full of rambunctious, playful, completely healthy mice. They had been given a chemical treatment known only as compound KL695. It was an azo dye linked to a compound called sulfanilamide, and it protected them entirely.
Section 5. The first miracle drug, the miracle of sulfa.
What truly cemented this chemical's legacy into the history books wasn't just a lab experiment on mice. It was a deeply personal, desperate story involving human life. Dr. Domach's own 6-year-old daughter, Hildegard, slipped and pierced her hand deeply with a knitting needle. A severe streptococcal infection set in, and red streaks began shooting up her arm. Her condition deteriorated so fast that doctors recommended amputation, but honestly, even then, death seemed imminent. Driven by sheer desperation, as any parent would be, Dr. Domach gave his daughter the unproven KL695 compound. Within days, her fever broke. She made a full, miraculous recovery. This was a monumental milestone in medical history.
But amidst all this celebration, there was a massive mystery completely baffling the scientific community. The drug worked absolute wonders inside a living host, like those lab mice or little Hildegard. But if you took the exact same bacteria and the exact same drug and put them together in a laboratory petri dish, absolutely nothing happened. The bacteria thrived. Why on earth did this wonder drug fail in the lab? Well, here is the massive twist. Discovered by French researchers at the Pasteur Institute, they realized that the living body's metabolism was actually breaking the compound apart once it was ingested. That azo dye, the supposed Trojan horse that IG Farben spent years and fortunes perfecting, was totally useless. It didn't do a thing. The active ingredient was actually just the attached tail, sulfanilamide. And get this, sulfanilamide was an unpatented compound discovered 25 years earlier. This shocking revelation completely ruined IG Farben's monopoly, but it was a massive win for humanity. It meant this life-saving sulfa drug could be manufactured globally, cheaply, by anyone who wanted to make it. To truly understand how dramatically sulfa changed the course of human history in just over a single decade, just consider the fates of 2 sons of U.S. presidents. In 1924, Calvin Coolidge Jr. got a simple blister while playing tennis. It became infected, and despite having access to the absolute best doctors in the world, he died. Just 12 years later, in 1936, Franklin Roosevelt Jr. developed a horrific, flesh-eating sinus infection, but this time his doctors used experimental sulfa. He made a full, complete recovery in a matter of days. The paradigm had officially shifted.
Section 6, Tragedy and Regulation, the birth of the FDA. But, as with all great leaps, we have to talk about the dark side of this boom. This incredible medical triumph quickly collided with the harsh, highly dangerous reality of the 1930s pharmaceutical market, a market that at the time was completely unregulated. Because literally anyone could manufacture sulfa now, companies rushed to market with wild variations, and they didn't have to test any of them for safety. One company decided they wanted to make a liquid sulfa cough syrup, which they called elixir sulfanilamide. But because sulfa doesn't dissolve well in water, their chemists used a solvent called diethylene glycol. It tasted sweet, it looked great, and they shipped it nationwide. The problem? Diethylene glycol is incredibly poisonous. Today, it's the main component of brake fluid. Scores of people, many of them young children, died agonizing deaths simply because companies could legally sell drugs without doing basic safety testing.
The public outrage over the elixir sulfanilamide disaster was deafening, and it simply could not be ignored. In 1938, this terrible tragedy directly pushed Congress to pass the Federal Food, Drug, and Cosmetic Act, which officially birthed the modern FDA. It established strict three-phase safety protocols. And these are the very same rigorous testing protocols that we all rely on today to ensure our medicines are actually safe before they ever hit the pharmacy shelves. So, from the ancient wild guesses of Theriac to the serendipitous discovery of quinine, the grueling grind of chemical dyes, and the accidental miracle of sulfa, our history of fighting infection is truly astounding. The discovery of sulfa proved once and for all that we could indeed kill invading microbes without destroying the patient, kicking off an absolute explosion in antimicrobial research. But it leaves us with a really compelling thought to end on. If progress took millennia of total guesswork before accelerating so rapidly into miracle drugs within a single century, what unimaginable breakthroughs are waiting just around the corner in our ongoing arms race against evolving microbes? Thanks for joining me on this explainer. Keep asking questions, and I'll catch you next time.